Welcome to the channel again. Time for disclaimer. Let's continue the gene series, and the topic is the operon. In a prokaryotic bacterium like Escherichia coli, shortly called the E. coli, there are two types of genes that includes structural genes which codes for the proteins and the regulated genes which controls expression of structural genes. Regulated genes can positively express structural genes and form proteins. Also, regulated genes can negatively influence structural genes and form no proteins. This entire package of genes that are sequentially grouped on the chromosome that can be completely turned on as a unit or turned off as a unit, this is the operon. Let's take lactose operon, also called the Lac Operon, as an example. Escherichia coli, when grown in a culture medium, and especially a medium that contains only lactose, E. coli expresses genes that forms enzymes utilizing lactose from the culture medium, and this is Lac Operon. Let's see the components of the Lac Operon. Lac Operon has 4 regulated genes and 3 structural genes. 4 regulated genes can be remembered by acronym RAPO, RAPO, R for repressor, A for activator, also called catabolite activator protein cap cyclic AMP complex binding site, P for promoter, and O for operator. 3 structural genes can be remembered by acronym ZIA. It includes genes LAC-Z, LAC-Y, and LAC-A. LAC-Z codes for galactosidase, LAC-Y codes for permease, and LAC-A codes for thiogalactoside transacetylase. These enzymes helps E. coli to utilize the lactose present in the medium and grow. Components of Lac Operon can be remembered as RAPO, ZIA. Let's see some basics of the components first before discussion. R stands for repressor gene. Repressor gene forms a repressor protein that can go and block the operator site. This prevents RNA polymerase to move beyond the operator site, so unable to read the structural genes. A stands for activator site, also called cap cyclic AMP complex binding site. Cap is catabolite activator protein. Adenylyl cyclase present on the membrane converts ATP to cyclic AMP. Cyclic AMP, when formed, binds to cap and forms cap-cam complex that activates the activator site. This stimulates RNA polymerase to read structural genes fast and vigorous to form more and more of proteins. P stands for promoter site, the site where RNA polymerase binds and initiates transcription. O stands for operator site, that can block the movement of RNA polymerase when repressor binds, or can allow the RNA polymerase to move further when an inducer relieves the block by repressor. Inducer can be a substrate isomer. It binds the repressor and prevents it from blocking the operator site. This process is much slower and less efficient without cap-cam activator site involvement, and the proteins formed remain nil or trace, if at all, without activation. Let's start with the first scenario, E. coli being grown in a medium that contains only glucose. And as glucose molecules enters the cell, adenylyl cyclase will be inhibited as ATP gets utilized in the glucose metabolism, and so there is no cyclic AMP. Repressor gene forms repressor protein that goes and binds the operator site and blocks it. RNA polymerase binding to the promoter site would be unable to read the structural genes. Lac Operon is turned off, there will be no gene expression and no protein formation. This is negative regulation of Lac Operon. Now let's see the second scenario, E. coli being grown in a medium that contains only lactose. And as lactose molecules enters the cell, repressor gene forms a repressor protein that tries to go and bind the operator site to block it. Lactose molecules becomes isomerized to allolactose that goes and binds the repressor protein and thereby prevents it from blocking operator site. This makes allolactose the inducer. RNA polymerase binds to promoter site and proceed to start reading structural genes easily. Further, adenylyl cyclase converts ATP to cyclic AMP. Cyclic AMP binds to CAP and becomes CAP-CAM complex that activates the activator site. Though inducer presence makes RNA polymerase to start the protein synthesis, CAP-CAM complex binding to the activator site speeds up RNA polymerase and thereby multiplies protein synthesis exponentially. Lac Operon is turned on, genes express proteins form that metabolizes lactose. And this is positive regulation of Lac Operon. Now let's see the third and final scenario, E. coli present in a medium that contains both glucose and lactose. And as both glucose and lactose molecules enters the cell, adenylyl cyclase would be inhibited as ATP gets utilized in glucose metabolism, and so there is no cyclic AMP. The repressor gene forms a repressor protein that tries to go and bind the operator site to block it. Lactose molecules becomes isomerized to allolactose that goes and binds the repressor protein and thereby prevents it from blocking the operator site. This makes allolactose the inducer. RNA polymerase binds to the promoter site and proceed to read structural genes easily. But as there is no cyclic AMP, activator site is empty. Though inducer allows RNA polymerase to start protein synthesis, the amounts formed are only trace. Absence of CAP-CAMP activation doesn't allow RNA polymerase to efficiently transcribe. Lac Operon is turned off. Little gene expressed proteins formed remain trace or nil. This is catabolite repression of Lac Operon. And that concludes the discussion on Operon. Knowledge knows no limits. Let you become limitless all life. And signing off for now.